Then about hospital learning about antibiotic policy guidance. Every hospital should have its own antibiotic policy. The antibiotic policy is essential for prophylaxis, empirical and definitive therapy. There should be periodic review and timely updating of the antibiotic. The primary aim of the hospital antibiotic policy should be to minimize the mortality due to antimicrobial resistant infection, preserving the effectiveness of antimicrobial agents. This can be achieved by selecting the patients who need to be treated, having all antibiotics available in the formulary, avoiding unnecessary antibiotic usage. The antibody policy should be based on spectrum of antibiotic activity, the pharmacokinetics, the pharmacodynamics, adverse effects, potential to select the resistance, cost, and special needs of individual patient groups. Every hospital should have their own antibiogram, and the hospital's drug and therapeutic committee should devise their antibiotic policy based on the there should be sample-wise antibiogram developed, and from each sample, the hospital should have the data of the isolated organisms and the antimicrobial sensitivity of the same. So there should be organism-wise antimicrobial sensitivity, sample-wise the clinical sample-wise antimicrobial sensitivity, as well as the department-wise antimicrobial sensitivity. The stewardship program should include a formulary restriction and pre-authorization, as well as monitoring in place. The goals of the stewardship program should be to have the right drug for the right person and the right time frame. The stewardship refers to a set of commitment and activities designed to optimize the treatment of infections while reducing the adverse events associated with antibiotic usage. The primary goal is to optimize the clinical outcomes while minimizing the consequences of antibiotic use, unintended consequences, toxicity, selection of pathogenic organisms like C. difficile, emergence of resistant pathogens, and secondary goal is to reduce the healthcare costs. About antibiotic policies, protocols. Now let us say about outbreak. What is an outbreak? It is an occurrence of infection at a rate greater than expected within a specific geographical area and or it is a curve. The epidemic curve is a curve that shows the progression of disease over a period of time. The shape of the curve helps in understanding the nature of the disease and its mode of transmission. So it's a we call it a point source outbreak when the outbreak when everyone falls sick within one incubation period. And the exposure time is brief and the epidemic course vanishes quickly. We say it's a continuous source outbreak when the outbreak involves ongoing source of contamination. Exposure time is prolonged and there is a longer and flatter and wanes only when all the people develop immunity. Then point source with the index case and a propagated spread. It starts like an infection starting from a single index case, but the newly infected people acts as a source of infection and there is a continuous transmission of infection. In such instances, only the outbreak stops only when there is a decline in the number of susceptible people. That was about outbreak and how to detect the outbreak by passive surveillance like monitoring the data, active surveillance by actively obtaining the data, sentinel surveillance from the healthcare data, syndrome surveillance that monitor the healthcare syndromes, and from social media and other newspapers outbreak. Now, after obtaining this information, the hospital should form and train the rapid response team, conduct periodic review of data, update the plan, identify the outbreak, arrange necessary drugs and materials, certain the diagnostics, and certain liaison with other healthcare settings. Outbreak investigation should include confirmation, description, and determine the cause and control of this. Regarding confirmation, the first step is to see whether there is outbreak and compare the current data with the baseline data and confirm their presence, which involves other causes such as increase in testing or lab errors or increase in population, and verify the diagnosis of the outbreak by clinical and lab methods. Describe what is the cause in terms of time, place, person, clinical and lab features. Have all the cases been found systematically? What are the description of the cases in terms of time, place, person? To describe the cases in the outbreak in terms of a graph, first in terms of first case, the response, the lab confirmation, and the potential cases prevented. In terms of place, it helps in identify the geographical spread of the disease and identify if there is a clusters. GIS, that the geography information system, may be used to map and track. And there's a place who is at risk in terms of age, sex, occupation, and ethnicity needs to be done. And evolved information in terms of time, place, and person, and pathogen, source, and transmission. Determine the cause with a hypothesis. Is the pathogen known? If it is not known, the conduct analytical study with by case control and code study. If the pathogen known, identify the source and the mode of transmission. For example, Vibrio cholera, contaminate what are the source, salmonella, the eggs or meat and others. Test for the hypothesis. If there are factors then determine the outbreak test for the hypothesis. Have analytical studies in place. Environmental investigations should include like contaminated eggs or appropriate food storage, hygiene, and other such data, including the laboratory data such as microbiology. And the control measures include behavioral controls, vaccination, then medications, environmental measures, health education, and inpatient control. Communication is an important aspect. Accurate and timely information regarding outbreaks should be communicated to people, internal and external to the organization. Communication reduces the anxiety, confusion, and misinformation, and plays a vital. Thank you.